Well, thank you very much for inviting me. It's really nice to be here to talk. And I apologise, I do have to explain what CILLIT Bang is. This is ubiquitous in Asia and Europe, and it's basically, you may have guessed, it's a cleaning product with a very cheesy advert. In the UK, we had this guy called Barry Scott who goes, kind of a big bathtub full of scum, and goes, and the dirt is gone. And it was very, very corny. It was just quite, it's quite well known at the time. And a much better looking guy in Spain. And here we have the Daily Mail's take on your average housewife, keeping it real as how it's all working. So, also I want to say that I'm Liz, not Elizabeth. Only my mother calls me that.
I'm so sorry.
That's okay. It's still kind of, I've usually done something wrong when I'm called that. So the dirt that I'm talking about, the persistent organic pollutants. So this is the plants that we hope to use in a CILLIT Bang way to clean up organic pollutants. They're horrible things. I think we all know they're resistant to environmental degradation. They persist in the environment. They can bioaccumulate in human and animal tissue and in the food chains and significantly impact human health as well as the environment. So that they're pretty nasty things, and sadly there's an awful lot of them out there. The Stockholm Convention in 2001 came up with a list of about 12 of the biggest offenders. Many of those are the first generation pesticides, and DDT is perhaps one that I'm sure everybody here has heard of. But there's also a lot of more recent ones, such as PCBs and dioxins, that are the products of manufacturing industries, particularly plastics and oil industries, that are now ubiquitous on the planet, all the kind of famous polar bear stories. So what I want to try and show you today is how we can use plants to clean up a persistent organic pollutant. And the persistent organic pollutant I want to talk about are explosives. So where do these come from? So they're not pesticides, they're not manufacturing from the plastics industry. These, of course, are from the military, and they're free sources. We've got the manufacturer here. There's a munitions plant in Birmingham somewhere, which I've no idea where this site is now. There's probably got some Barrett houses on it, and they're all happy. Here's one up in the north of the UK, which the roof has collapsed on now and has just been fenced off, and it's not really known how to clean this up, what to do about that. And then a big source of pollution as well is in the use of explosives when a big shell or a round is detonated on the range. You get quite a lot of residues here that remain inside the shell, and they percolate out and go into the soil, and you get a lot of pollution from that way. And finally, there's decommissioning. There's Obama looking at some decommissioned shells. That's all jolly and good. And here the squaddies have stacked these bombs have sell-by dates. You know, thought about they can't use them after a certain time. So they stack them around the back of the mess tent, and they leach into the drinking water slowly. So there are lots of different ways that we can get this pollution. So TNT is the big offender here that I'm going to talk about in the first part of my talk. Here's the structure. It has these free nitro groups which pull away the electrons from the ring in the middle there and make it very difficult to degrade, actually mineralize this compound. It's extremely toxic, and here we've got some workers in a munitions factory in the First World War filling shell cases by hand. This causes liver dysfunction, and it's a carcinogen as well. So it's really, really horrible stuff. But it's okay because they've got a bonnet and a pinny, so there's no worries, they're going to be fine. The scale of the problem as well, it was enormous. We've got 10 million hectares as an estimate in the US, and that's alone. So globally, it's hard to estimate, but it's certainly out there. A particular problem with TNT is that it binds very tightly to the humic content of the soil, so it's not very mobile. Once it kind of sticks here, it's a bit like kind of Velcro stuck to the surface of the soil. And here on your bottom right, we've got Arabidopsis plants growing on a range of TNT-contaminated soils, up to ranges here that are found on military training ranges. So these are levels that the plants are encountering out there, and you can see that Arabidopsis is certainly struggling, and it's dying at the higher concentrations. So how to tackle this problem? There are a number of existing cleanup strategies that are all commendable and lovely, but it's just one heck of a landfill hole you're going to have to dig to put 10 million hectares of contaminated soil in. So it's basically just the scale of the problem. We can't really achieve it using the current strategies. So I'd like to present to you today phytoremediation, this wonderful green area in the corner here where plants are going to do wonderful things. I say low maintenance, but you haven't seen my garden. It's minimally disruptive, cost effective on a large scale, and everyone loves plants. Everyone here, I've got a committed audience, so that's what we're going to try and do. So to understand more about how plants detoxify as antibiotics, we want to look at the biology behind the plant's response to TNT. There are the 4 well-characterized methods of dealing with this compound when it comes in. So there's an activation step, a conjugation, compartmentation, where you would stick it into the vacuole or slide it into the cell wall or under the carpet. And transformation occasionally happens as well, where you may sequester it outside of the plant, for example. So the first step here, activation. We did a microarray a number of years ago, and we isolated some genes that were upregulated in response to TNT. And the oxophytodienoate reductases came up. Now, these genes are strongly upregulated in response to TNT. So we took these genes, cloned, expressed, purified, and we found that they did have activity. The main pathway is along the top here. They have this secondary pathway here, but it's a minor component. Basically, here's a TNT structure, the nitro group, and that's converted to a nitroso, a hydroxyl amino, and an amino group. And then we go on, we functionalize the molecule, and we can then go on to conjugate it. So if you overexpress these OPRs in plants, then you increase the resistance to TNT. Similarly, if you knock it out or down, then they're more susceptible to TNT. So the second step is the conjugation. You're going to stick a sugar molecule onto that. So we found some glucosyl transferases with the help of Diana Ball's lab and characterized those. And we found they had 2 different ways of conjugating to the activated molecule here. They've got a C-glucoside link and an O-glucoside link, and they'll conjugate to the hydroxyl amino and the amino. So you get a whole swathe of conjugated products, which can then we're not sure exactly where they end up, but it looks as though at the moment they're going into cell wall material. Again, if we overexpress these, we see an increased resistance to TNT. Ooh, a couple of pops. And then there's a second group of conjugating enzymes, which we didn't really get around to until more recently, and I wish we'd done this one first. We found 6 Arabidopsis GSTs that are expressed and purified and have activity towards TNT, and we isolated these 3 conjugates. So you have the TNT molecule there, where the methyl group is conjugated, the sulfur for this one and this one. Because this one was the one we were really whooping about with excitement. As this conjugate free here is concomitant with the release of nitrite. So what we've done is, with this, it's very laissez-faire attitude of this enzyme, is very readily cleaved this nitro group straight off, producing a dinitro compound, which opens up this molecule for subsequent mineralization if we're lucky. We've destabilized the ring, we've produced something that's much more amenable to degradation later on. So we, very recently, and this is work that Kyriakos in the audience down here holding the microphone has done. He has a poster, a P14, if you can see it. When we overexpress these lines here, we've got a couple here, on agar plates containing TNT, we can see we've got longer roots than the wild-type plants. And when we do soil-based studies, and we grow the plants for 6 weeks in soil contaminated with 2 different concentrations of TNT, these are the expressing lines here, and they all have less TNT and less ADNT than the wild-type line after 6 weeks. We don't get all the TNT out. Some of it's stuck like Velcro. It's quite hard to get back out of the soil, and some of it's lost. So how to increase the TNT detoxification in plants? We could take these endogenous enzymes and upregulate them, but it doesn't really give it the CILLIT bang we're after. So what we did was we took some bacterial nitroreductase from some TNT-contaminated soil. And when you put this enzyme, express this in Arabidopsis, it's doing the same step here. It's taking the nitro group to a hydroxyl amino group, but it has a massive effect on the resistance to TNT. So here we've got the wild type there growing on TNT, and it's really struggling. It's not going to make it to the next generation, whereas the nitroreductase expressing lines are setting seed, and they're doing very well. So that's increasing TNT, but TNT is not alone. It has a kind of Bonnie and Clyde relationship with this other chemical here called RDX. So this is hexahydro 135-trinitro 135-triazine. It looks like this. It's packed into all the shells with TNT in pretty much a 1-to-1 ratio, and so if you find TNT on the range, you find RDX. Again, there's been little degradation in the last 60 years, and it's toxic. And unlike TNT, which sticks to the surface, the RDX percolates very readily through the ground, through the soil, and into the groundwater. It's more like a kind of a Vindaloo curry effect. So if you want, and this is now causing real problems for the US Army. And here in Cape Cod Peninsula, they're mapping plumes of RDX that they're trying to clean up. So if we want to clean up this RDX, we need plants that can survive in this toxic environment with the TNT. TNT is more phytotoxic to the plants. So we combined, the problem. So yeah, there's Hank and Marv on the training range there with their weapons. A little bit of rain comes along, and the RDX leaches into this sole source aquifer underneath Cape Cod, and then all the drinking water has to come from here as well. It's not just RDX that's been detected in this aquifer. There are a number of other horrible, other persistent organic pollutants as well. But there are worrying statistics based on the water that's been sampled from this. And the Environmental Protection Agency in the US have stopped the use of live ammunition training on this range because of the RDX that's leaching through. So it's a very important problem that the US want to address. They want to clean this up. So to try and work out how to clean up the RDX, we took the same approach, and we went back to some land here that's military land in the UK. We took some samples. Here's the Army were very happy to demonstrate how they could blast a big hole from the earth. We sent the French postdoc down to get the samples. So he came back, and we isolated this Rhodococcus 11Y from some RDX contaminated soil, and the Rhodocrus, obviously the name is a lovely reddish-orange colour there. And this wonderful P450 was isolated, which is a unique structure. And I don't have time to talk about that, but the references are here, and it's a really lovely story about how that works to break down the RDX. And the best thing about this is that it catalyses the first step, which then leads to the subsequent mineralization of this compound, which is a real holy grail in the world of persistent organic pollutants. So if you combine the nitroreductase that's detoxifying your TNT with your RDX degrader, then we have some fantastic results back. So here we've got plants all growing on uncontaminated soil, and here we've got soil with TNT and RDX in. So the wild type plants here, they're all dead because of the TNT, basically. They can't survive. The TNT detoxifying plants, the nitroreductase expressing ones, they're doing quite well. They're probably set some seed. The RDX degrading ones are struggling again because of the TNT. But if you put them together, you get this really brilliant effect whereby they're able to withstand the toxicity of the TNT and use the RDX like a fertilizer because it's full of nitrogen, and they do really well out of it. So I'm really chuffed with that. So then we tried to mimic Hank and Marv on the training range in a pot of Arabidopsis. So we took a Arabidopsis grid in a pot, watered in some RDX, had TNT in the pot, watered in the RDX, let it sit for a week, and then flushed it through with water to see how much RDX was left after 7 days. And you can see here, well, either when they're grown in uncontaminated soil or in soil with TNT, in either case, our triple transgenic lines here have removed nearly all of the RDX within that week. Whereas the wild-type plants here, the light blue, there's still the same amount coming out of there. It's a little bit lower here, but there's still a lot more here than in the transgenic lines. So that looks like it's going to work in the field. But we just thought we'd do a couple more tests on these plants. So they've got more nitrogen from the RDX. What's happening to the levels of RDX in the leaves? Well, the wild-type plants have got a whole load of RDX in the leaf. That's predominantly where it accumulates in the plant. Whereas our magic lines have got much less. They've got 30 to 100-fold less RDX in the plants. We then spoke to Sue Hartley and did some herbivory studies, because it transpires that where I like donuts, herbivores like locusts like high nitrogen plants. So we thought we'd better test that they don't prefer these plants over these, because they do have more nitrogen in them. And there's no difference in herbivory in this instance, so that was good. Also, the graph on your right here shows that our plants have got much more biomass when they're grown on soil containing RDX and TNT. So our plants are growing bigger, and they've got less of this toxic pollutant in them, which should have impacts on the food chain as well. But Arabidopsis is not the plant for the Army. The Army like to grow, they like to drive their tanks over things, and it's quite, Arabidopsis is not going to take that kind of abuse, really. I love the way they pose as well. It's good. We drove over a car. Yeah. We spoke to our collaborators in the US. So, well, what would be better? And they said that they would like perennial grasses. Wheat grasses we tried at first, and we have moved on now to switch grasses. But at the time, the model system to use was creeping bent grass, just to show if this technology would work in a monocot. And here are our transgenic RDX degrading grasses on the right-hand side. And you can see as well the biomass effect of the nitrogen they're getting. And you can see here our unmodified grasses have not had any impact on the RDX, whereas the transgenic lines are doing very well. So then we wanted to transfer this technology to the grass species. There's another car being driven over another guy. Yeah. So that this is the plant of choice. All the recent work in biofuel crops has made switchgrass protocols for transformation very, very good now, and we were able to use those protocols to transform our genes into switchgrass. They provide year-round cover, and these guys they literally blow this stuff off the face of the planet, and then it lands back down a big plot, and then it has to start growing again. And switchgrass is apparently quite good at that. So it's perfect, and we've been able to transform it, and we've got the plants growing now already. So we're starting field trials. We've got 5,000 to 10,000 of these plants going up now, ready to go in late spring 2014 to see if we can actually test this GM in the field and see if it really works. I think I've got a minute to just explain the last experiment here that our collaborators in the US were doing. It's not like you can just go out and dig holes and plant these plants on the training range, because the training ranges are littered with unexploded ordnance, and you wouldn't even be allowed on there. So they'd have to seed these using helicopters and drop the seed out. So they can't just drop the seed because it's not very likely to take. They need to give the seed a little bit of a nutritious environment to support early growth. So they developed the seed ball strategy here, where they mix them kind of fertilizer and soil and a kind of clay base with the seeds, and then they were going to drop them out of the helicopters. So before they got to that stage, they started dropping them from ladders. Then the ladders would, the helicopters would never be that low to the ground, and so they went outside and dropped them from the roof. So yeah, okay, that's keeping it real. So we're hoping that we'll be able to take our RDX degrading TNT detoxifying transgenic grasses, perform this field trial, and then ultimately, if we're really lucky and this technology pans out, we'll be able to clean it all up, which would just be wonderful. Make my life. I'd be really chuffed with that. And finally, acknowledgements. Of course, Neil Bruce is sitting at the back there. This is all Neil's work. Thank you, Neil. Our collaborators in the US with the seed balls is Tim and Tony here. We're doing that work. And Stuart Strand, a collaborator in the States. The BBSC, CERDIP, and DSTL have all been instrumental in getting funding towards this. And just quickly draw your attention to posters. Emily Bainan, P10, is talking about why TNT is toxic to plants, which we now know. I've already given Kyriakos a good plug, but we've got a visiting student, Charit here, poster number P13, and Zakran P12 as well, on different subject matter, but still really, really interesting if you've got time to see their posters. Thank you very much for listening.